From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on October 23rd, 2023. Hello, Micronauts, and welcome again to today's Quality Quorum. I'm your host, Dr. Mark Martin of the Biology Department of the University of Puget Sound in Tacoma, Washington. Today is lucky episode 13 of Matters Microbial. Thank you very much for listening and or watching. First, today's denizen of my Wunderkammer. A wonderful crafter in the UK invented the positive potato, so I convinced Hooked by Bex to make me a gram-positive prokaryote with an empowering message. It's true that microbes can survive anything, so can we. Bex is a wonderful artist, and I'll put a link to her Etsy shop in the show notes. As for today's inspirational lapel pins, yes, Halloween is approaching, and I adore all things mysterious and creepy, like cryptids. So when Bigfoot only leaves footprints on this pin, it's doubly funny to me. The second pin is more serious and very close to my heart. It is so nerve-wracking to learn new things. Very few of us start out doing a good job at something new. I know I'm not that kind of person. But trying is the only way that we grow. As Winston Churchill said a long time ago, never, never, never give up. I not only teach microbiology, but I have a small lab staffed by undergraduates. They call themselves Doc Martians, which makes me smile. Here is my current crop of wonderful students, wearing hats, of course, in our lab lunchroom. And here they are, illuminated by bacterial light. I just adore this kind of photography. It starts out with a single bright flask, as you can see here, and is followed by an incubator filled with bacterial bioluminescence. I'd like to take a moment, if it's okay, to enhance your OMG, your overwhelming microbial greatness, with a special bacterium. I'd like to introduce you to the fascinating gram-positive bacterium Panabacillus. You may have seen the almost artistic colonies formed by this bacterium on solid media. Here's a really good example, and there are many others that you can find. This kind of imagery led a former micronaut of mine, Megan Hadasaka, to create this wonderful wood block print of the colony appearance. It's true, I have a lot of student art up in my lab. However, when I try to stain those Panabacillus colonies, my results are, as you can see, not so impressive. But during one of my soil safaris that I do with students to look for possible antibiotic producers, I saw this very unusual soil plate. It looked like Panabacillus. When I carefully restreaked it, it was even more like that. We went on to actually do 16S ribosomal RNA identification, and indeed it was Panabacillus. The swirls and patterns are just fascinating. I've put some links in the show notes for both new and experienced microbiologists. There's so much to learn and about which to marvel in this world of microbiology. Remember last week's listener-viewer question? It's about the number of cells to be found in a bacterial colony. Well, new Doc Martians Ariana and Ruby decided to follow up. We decided to repeat last week's experiment, except when one of them used a big colony, as you can see here, and the other used a tiny colony, as you can see. Would they get different results? As we expect. The plates glowed beautifully as seen from this, and the tenfold dilutions you're looking at now, they kind of tell us that both Ruby and Ariana have well-honed microbiological skills. This is one reason I really adore using bioluminescent bacteria to teach basic skills in microbiology. In any event, the tiny colony had 1.42 times 10 to the sixth cells, while the larger colony had 9.2 times 10 to the seventh. Now, this might seem simple, but it's actually a great experiment and experience for new micronauts, teaching them skills and analysis and bringing up other questions. For example, do all bacterial colonies follow this kind of rule? How much of a colony is made up of cells, and how much of it is made up of matrix? And as you can see, Ariana is Doc Martian style happy about her work, and so is Ruby. I'm very, very lucky to have such students in my lab, and I simply can't wait to see the kinds of projects they'll have when they get ones of their own. The ninth week of my microbiology class continues. As we explore how to measure antibiosis, we're learning that the background bacteria concentration is important. Briefly, we spread bacteria on a plate and then challenge those bacteria with spots of other bacteria that we think are making antimicrobials. If such compounds are being made, zones of clearing will appear where the background bacteria cannot grow. As you can see with this image of micrococcus, using diluted background bacteria gives a much clearer view of what's happening with clearer zones of inhibition. This may be due to how quickly the background bacteria grow, reducing the impact of the antibiotic. What this means is that we need to fine tune our assays one background strain at a time. I have to tell you, a very wise friend of mine once called this the re in research. Moving on, I don't know anyone who doesn't find magnetism fascinating. I certainly do. So imagine my surprise on a visit to beautiful Muriwai Beach in New Zealand a few years ago. Not only was it quite a beautiful scene, as you can see, but the sand was composed of magnetic iron particles. When I put a bar magnet under a sheet of paper and pour this sand over the top, you can see the field lines quite clearly. I've included some links in the show notes to this phenomena. I'm told that this magnetic sand is due to volcanism, but I can't help suspecting the small masters have something to do with it. And in fact, there are some bacteria that navigate using tiny internal magnets. I will include several links in the show notes for both beginners and experts. And who better to tell us about this wonderful phenomenon of magnetic bacteria and the role of compartments in bacteria than Dr. Arash Komeili of UC Berkeley's Department of Plant and Microbial Biology. Welcome to our Quality Quorum, Arash. It's so nice to see you.
Great to see you, Mark. Thanks for inviting me.
Oh, it's such a pleasure. Your work has been much on my mind.
Thank you. Yeah, this magnetic sand has taken me down a rabbit hole as well, trying to understand what it is. But you, you can see how easy it is to get distracted with just the fun of it, and I think that's part of what defines a lot of our work is that it's both fun, but at the same time, is start getting into the details of how microbes are making magnets, then it becomes quite fascinating.
I agree. Can you give a little bit of a background to people?
About myself or about the bacteria?
Let's start with you.
So yeah, my name is Arash Komeili. I've been a faculty member at Berkeley for almost 18 years now, and the almost the entire time I've been studying these magnetic bacteria. Um, my background really was not at first in microbiology, or it was in microbiology, but when I got interested in pursuing research as a PhD student, I was more fascinated by how cells organize themselves. And what I was told and taught was that if you are interested in that problem, you don't want to study bacteria because there's really not much going on in there. And so, that's what I pursued as a PhD student. But as a for my postdoc after my PhD, I was interested in working with bacteria again, and through some good fortune and luck in just conversations with people, someone mentioned that there are bacteria that have organelles, and that just kind of blew my mind. Particularly because I was taught explicitly that they don't have organelles. And to this day, I see it in textbooks that they don't have organelles. And so that really led me to think about it more and ask more questions. And I found out that the compartments are from bacteria that make magnets. And so that's been a topic of study for me since 2001. So quite a long time now, 22 years.
It's really wonderful. I'm really resonating with what you've said. The number of times that people who maybe took a microbiology class some time ago and haven't really kept up will tell me that such and such is always true, such and such is never true, and the microbes could care less.
Yeah. Yeah.
They, I have a saying: "First evolved, last extinct."
Right.
And it's really true. So, I mean, while it's easy to look at as a look at a eukaryotic cell like a bento box of different locations, I always thought that surely there's levels of organization inside of a bacterium. We just lack the ability to perceive them. And even when I was in college learning about these things, then people would say, well, gas vesicles don't count. Well,
Right.
why not? Or carboxysomes don't count, or ammoxysomes aren't real, or
Right.
whatever, fill in the blank. The simple fact of the matter is, um, all eukaryotes pretty much owe the basic strategies, let's face it, to bacteria and archaea. That's what we're dealing with here. But
Yeah,
I
I
am a
think
microbial centrist.
right. No, I don't disagree, and I think there's a lot of value in doing the compare and contrast, right? And maybe even taking a step back, thinking about why do we want to say that bacteria have compartments or don't have compartments, or make organelles or don't make organelles. A lot of these hard and fast rules, textbook rules, came about particularly because people were looking at microbes and they really wanted to be able to distinguish them from each other, right? So if you saw two photosynthetic microbes, how do you know if one is a bacterium and another one is a eukaryotic algae? They're both microbes, but they clearly look different. Or if you saw something very intracellular organism, is it a virus or is it a bacterium living inside another cell? So right before basically the sort of modern DNA-based approaches for identifying organisms, people started using cellular organization as a way of categorizing different organisms, right? And a simple distinction between very complicated looking eukaryotic cells and simpler looking bacterial cells was this organization. And I think at that time people were much more very specifically looking at big things like, is there a nucleus, is there these very obvious big things that are where the DNA is compartmentalized. Not to say that bacteria can't have cellular complexity. That wasn't the intention of the people who came up with the distinction, but that's just how it evolved throughout years of people just looking at bacteria as not being very interesting or really good for studying biochemistry, or as a sort of bag of enzymes. But even the simplest bacterial cell, something like an E. coli that doesn't necessarily have compartments, it does separate its periplasm from its cytoplasm. It has to know to put flagella in certain places, has to build those flagella. So even the basic questions of how do you know where something is in the cell, or how do you form, chemically different compartments, any bacterial organism has to do that, right?
Well, it's so funny to me because as I'm teaching microbiology and we were talking about how the FTSZ protein is able
Yeah.
to localize to the center of a rod-shaped cell, and they'll talk about the nucleoid occlusion model.
Right.
And then the student looks at me and says, "But how do the min proteins know to be at the poles?" And then they say my eyes get all glittery because I
Mm.
think it's so cool. And this is because I think all of us want to simplify things and put them on bumper stickers almost. And so I have what I call the three deadly centrisms. And the first is oxycentrism, the idea that oxygen runs the show.
Hmm.
And it's a real challenge with my microbiology students to get them thinking about other electron transport chains.
Right.
Secondarily, eukaryocentrism. They're so fixated on eukaryotes. The number of times they'll insist to me that bacterial flagella wave, right? Because they're used to that 9+2 model when they've generally seen Chlamydomonas
Right.
as the example in class. And then finally, and this is the one that I think is super relevant today, I call this coli centricity. And even microbiologists will start to think that all bacteria are like E. coli, and it's simply not true.
Right.
And
Yeah,
and
I agree with you. And kind of going back to the idea of using eukaryotes as a model, and then let's say we even accept that bacteria have organelles. The first way that we think about it, and I'm not actually pointing fingers at anyone because I used to think of it this way, is, oh, wouldn't it be cool if you could find the bacterial equivalent of this thing that happens in eukaryotes, right?
Mhm.
But actually, time and time again, what we find is that there are actually just different strategies that bacteria use to make something that looks the same. Even kind of thematically, it might be the same, but it doesn't necessarily have to have happened with the same, using the same proteins or even relatives of the same proteins to do the same thing. So when I was saying, like, why do we want to study organelles in bacteria, I think part of it was, let's think about kind of some reasons beyond, oh, we didn't think they had them, now we know they have them. Isn't that cool? But thinking about what are the challenges that it brings up for a cell, and how does it solve those challenges. um, can we use the organelles for something? Starting with the organelles, can we learn something about some biology that happens in the environment that we didn't know about? And I think those are more interesting questions. It might still get us to the point where we realize, oh, similar things are happening in bacteria and eukaryotes, but it's equally likely that we'll discover something totally new, and either answer is interesting, right? So it's been hard, I think, for the field of bacterial cell biology. I would say the last 20, 25 years, it's been really picking up steam. And I think it's been hard for us to do two of those three things that you said: separate ourselves from having to find eukaryotic equivalents in bacteria, and then start thinking about models that are different than E. coli. And I would put in there for our field bacillus, because those are so two really powerful ways of thinking about things. And one of the nice things when you go to conferences recently is seeing other really interesting microbes kind of take the center stage for studying cell organization, because some of them have more, microbes are masters of efficiency. So it makes sense that not all of them have the same organelles. They just have the ones that are useful for the things that they need to do. And so you really need to go and explore new and different kinds of microbes to see new and different types of compartments.
I tend to use, I mean, when this first started becoming an issue in pedagogy, I would have people argue with me that a compartment that was bounded by proteins is not really an organelle, and I
Right.
would come back and say, "Well, what is the function of an organelle?" I felt like I was doing some Zen thing, but it's really true. I mean, you need a different set of conditions. So when I talk about carboxysomes, I talk about the conditions the enzyme Rubisco requires, having relatively low concentrations of oxygen and high concentrations of CO2. This is the same principle that chloroplasts, which were originally bacteria, have to deal with. So that's what it comes down to, rather than what the structure of the compartment is.
Right. Right. I mean, if we kind of, like, historically, they were considering membrane bounded compartments that had nucleic acids in them. That's sort of what
Right.
they were talking about when they talked about organelles in eukaryotes. So, some things don't count if you want to go that way. But the definitions become so fuzzy now that, in eukaryotes, they talk about membraneless organelles now, right? Where it's just phase separation, creating like a new kind of environment within the cell. And so they talk about it as organelle formation.
Yeah.
Uh, and some organelles in the eukaryotic cell have shared either ancestry or mechanisms of communicating with each other or building, but some of them are very different. So it's the term organelle now has devolved into meaning things that are in eukaryotes but not in bacteria, right? It doesn't have a useful definition anymore.
I
If there's
agree.
something in eukaryotes that we want to call an organelle, it's an organelle. But if bacteria have a compartment, and if you don't want to talk about protein bounded compartments, there's plenty of lipid bounded compartments in bacteria,
Right.
and so then it's not useful anymore, right? So for our work, at some point we decided to kind of have a simplified definition that can at least guide you towards understanding how compartments are formed. And if the answer is the same as eukaryotes, it'll be easy to check. If it's different, it's going to be obvious as well. And so, we're thinking mostly about lipid bounded compartments, and to take some cell membrane and turn it into a compartment, there's a lot of challenges involved in that. And so, those challenges are solved by very specific types of proteins in eukaryotes. So we think about a compartment or an organelle, lipid bounded organelle, as something that has a membrane separated, or distinct part of the cell that's separated by a membrane. It has a unique group of proteins, and those proteins in the membrane allow you to do some function that you can't do very well if everything was just floating around in the cell. And it might be that, kind of like Rubisco, you need it concentrated, but it might also be that some intermediates of that reaction are toxic. So you want to keep it away from the rest of the cell, right? So you're making a factory, but also you're protecting the surrounding environment from what the factory does. And it makes everything more efficient. And so now, we're studying a compartment. If it doesn't happen at all in eukaryotes, or the way that it's made is not at all the same, is still really interesting to um, identify it, figure out how it works, and then figure out if we can use it for other things other than just understanding the bacteria.
I think structure and context is everything. And it doesn't surprise me in the slightest that there has to be internal structure. I think that we lack some of the technology to see some of that internal structure.
Yes,
And now with new techniques, and I saw some of the beautiful images that you're going to be talking about in a moment, we can start to see that, and it's beautiful structure all the way down.
exactly. I think just the advances in imaging and in microbiology now, there's just so much more focus on diverse microbes, and we can both culture different kinds of microbes or just study them. But at the same time, even if we can't culture everything, we have access to so much more comparative sequence information. So you discover something in one case, you can easily go and see how common is it, or are there things that are similar to it just based on sequence. And in terms of imaging, now using for example, cryo-electron tomography as a way of, with minimal perturbations, seeing a structure within a cell is really incredibly powerful. I
It's
don't think we could do most of what my lab has done without those advances.
It's so beautiful. And we recently had Ariane Breigel on the
Yeah,
podcast to talk about the techniques, and I cannot emphasize how beautiful some of the internal structures are. And that,
right.
but that's me. I guess I want to be the poet of the microbial world.
Yeah, and you are. You're succeeding.
Well, I did want to say because, and I'm glad that you talked about compartmentalization because that's a big interest of mine as an instructor.
Mhm.
And I think you've given me lots of things to think about, especially those new lipid phases that are thought to act almost in the ability to create different compartments in cells,
Right.
which I don't quite understand yet, but I'll get there.
Yeah, and there's quite a bit of evidence that um, a lot of those types of phase separated compartments exist in bacteria as well,
Hmm.
that are really important for even things like potentially how a carboxysome is built. It might need to rely on that type of biophysical interactions to do it, and also other compartments. So I'm happy to answer questions off the air too about that.
Oh,
Yeah.
yeah. Uh, so I guess we should move to our magnetotactic bacteria. And by
Yes,
the way,
my babies.
shout. Yes, but a shout out to a friend of mine who I met at the University of Michigan long ago, Marion Schmidt, who
Yes,
has magnetotactic bacterial jewelry.
yes. Marianne, if I'm not wrong, either as an undergrad or a postbac, worked with Dennis Pazelinski on magnetotactic bacteria. So I had I think I met her long, long time ago when she was still an undergraduate or before graduate school at an ASM. And she did really wonderful work. And so it's great to see her kind of moving up and still holding on to her love of magnetotactic bacteria.
Oh yeah, I believe there's a bolo tie,
Oh wow.
and and that's an image that we're going to put up. And then in addition, the earrings, but they couldn't find those for me to put them up there. But
Wow.
Marion, applause at Cornell now.
Yes, yes. Great.
So the first thing that I want to ask is that the discovery of magnetotactic bacteria only took place, I want to say, in the 1970s. Am I wrong about that?
No, you're right. The sort of the first official discovery that led to a published work, and it was an accident. It was by Richard Blakemore, and he was doing some work at Woods Hole, and there were some samples of mud from Salt Pond, Woods Hole, and then he was looking under a microscope at some of those samples, and he noticed that there were bacteria that had kind of similar morphology, but they were all swimming in the same direction. And so, this is 1974, 75, and he didn't know what the stimulus could be to have them all go in the same direction. I think he first thought maybe there's light coming in from the window or something like that. And he just moved the microscope to different parts of the lab, did various changes to the setting of the experiment. And what he noticed was that these bacteria are always swimming in the same geographic direction. And so his thought was, okay, what can tell an organism what the geographic direction is? And then he thought about magnets. He brought a bar magnet next to the microscope, and he could override their swimming and change the direction of swimming. The other big thing was that he was able to concentrate enough of them to do some electron microscopy. And when you do that, boom, you see these little, very dark, electron dense crystals within the cell that are made up of iron and oxygen in that case, called magnetite. And so that was, I think the paper is a solo author paper, and it's called Magnetotactic Bacteria. I think that's the name of the paper. Now, what's interesting is, as a field, sometime in the early 90s, we figured out that that is not the first discovery of magnetotactic bacteria. And in fact, there'd been a medical doctor, Bellini, in Italy. He was a medical doctor, and after World War II, he was working on a project to look at the effect of sort of metal contamination from various bombings and things like that in water. And he noticed, looking under a microscope in the 60s, 50s, and 60s, he noticed what looked like bacteria that were all, again, swimming in the same direction. And he did a series of really beautiful experiments to try to, without culturing them, to understand the physiology of these bacteria. In fact, he was so worried about the metal in his microscope affecting that. I think he built a microscope using almost all wood just to make sure that he wasn't impacting the bacteria from the microscope. And so he discovered these things. He wrote a couple of papers for the university sort of internal dissertation system, and the committee looked at it and they didn't believe him, so they didn't allow him to publish it. It was published internally, all in Italian, so there was a record of it, but it was never published outside of that university, so nobody knew about it. And Richard Frankel, one of the sort of really incredible people in our field, discovered this, and he tracked them down. He tracked down those papers. They translated them into English, and they republished them.
Oh,
So
that's wonderful.
yeah,
Yeah.
so, and I think he was able to see that process through before he passed away. So he saw that he got the credit for his discoveries before that. So uh, you wonder how many other people over the years, maybe even going back to Leeuwenhoek or someone else, looking under a microscope, saw some bizarre behavior like that and just couldn't figure it out. But you know, Blakemore
So
was in the right place at the right time. And I think, like we were talking about technology, having the electron microscope really made it so that the behavioral experiments are one thing, but when you see that bacterial cell that has something extra in it that is so unusual, then those two together are much more convincing.
so you no doubt we have some images that viewers can look at
Yes.
of some of the bacteria that have what look like chains of crystals inside them.
Right.
And each of those crystals is what?
Each of those crystals for your typical magnetotactic bacterium is made of magnetite, which is Fe3O4. It's the same mineral as lodestone that was used to build compasses for navigation by humans. And what we're looking at there is that each crystal in the electron microscopy image is something 50 to 70 nanometers across. And it's a single crystal. It's not multiple crystals that are fused together. It's a single crystal and it has a permanent magnetic dipole moment. That really means that it's very, very difficult to change the north-south direction on that one little magnet.
Wow.
But there's a bunch of them next to each other, and they all have their north-south pointed in the same direction. And that dipole moment is additive. So the cell has a long kind of compass needle in it. And that compass needle will try to align with external magnetic fields and is sensitive enough to align with the Earth's magnetic field, which is many times weaker than like the stir bars we use in the lab or fridge magnets or something, right?
Well, this
So go ahead.
I was going to say this is why you have many little crystals,
Yeah.
is that they can form, for lack of a better term, that compass needle that you mentioned.
Yes.
But why are they attracted to those field lines?
So that's a open question. I can give you the best uh, theory in the field or best hypothesis. The idea is that in most aquatic environments, and these bacteria are really, really cosmopolitan, you can go almost anywhere in the world to sort of the water sediment interface, and you can find magnetotactic bacteria and many, many different kinds of magnetotactic bacteria. If we did what you were mentioning earlier, doing the 16S ribosomal RNA, there's not a ribosomal RNA sequence that will tell you, oh, I have a magnetotactic bacterium, because they just are spread all across the bacterial tree. And so in those environments, some things are predictably some nutrients that bacteria need are kind of very predictably organized. So oxygen, for example, is high at the top and low at the bottom, right? It doesn't dissolve very well into water, and there's organisms at the top that are using it. But all of those organisms at the bottom that are anaerobic, they don't see it. And some of the things that they produce at the bottom during respiration counteract the oxygen and get rid of it, right? So there's opposing chemical redox gradients, and those are sort of layered top to bottom. And in most places on Earth, the magnetic field is cutting through those gradients, right? So it's almost like a little roadway through the high to low oxygen or high to low sulfide or something, right? And these bacteria are using your chemotaxis or aerotaxis mechanisms that all bacteria have, swimming along and trying to figure out where it's best for them to be, for if they need a certain amount of oxygen or if they want to avoid high oxygen. And to go from point A to point B, they're restricted to these roadways that are the Earth's magnetic field, and it makes their search much simpler. That means they take fewer steps to go from A to B than your bacterium that's kind of exploring three-dimensional space, right? So the idea is that it makes swimming more efficient. Basically, you don't have to use your flagella as much, which means you use less ATP, and maybe that's the advantage, right? That's the idea. And there are some experiments in the lab to support that. For example, you can set up oxygen gradients, and you see that magnetotactic bacteria will go to a particular place in that gradient. If you add oxygen, more oxygen at the high end, this band of bacteria will relocalize to get away from it, right? And now you can set up that same experiment using either bacteria that have magnets or mutants that don't make magnets, or giving them a magnetic field to guide them to swim along this oxygen gradient. And you can see that the running away from oxygen is much faster if they have a magnetic chain and a magnetic field direction to guide them. So there
That's
is,
so
some experiment to prove that they can do aerotaxis more efficiently.
So is it true that anyone who is like a listener or a viewer could just gather up some muck from this and that kind of the in like a pond and stick a bar magnet to one side and kind of start looking for these?
Yeah, I think the trick is that you need a microscope, I think, in my opinion.
Yes.
That's kind of a necessity because and your magnetic sand reminds me of this. Magnets are not that hard to make. So in the muck, there's a lot of iron that's turned into magnets from just non-biological processes. So if you just put a magnet, you will see something stick to it, but it's very likely that that's not just bacteria. So seeing something next to your magnet does, you won't be able to see it. So the way that we do it is, often you collect some samples and you let it just sit around for a little bit to increase the numbers of bacteria in there. I think it's mainly that, you bring them from a cold creek to room temperature and things can start growing a little bit. But you can even start with just a sample that you take right away. You could put a magnet next to it to kind of get things to stick better. And then you collect that drop of water. As I'm sure your listeners uh, who are a little bit more familiar with microbial diversity, that drop of water will have lots of different kinds of bacteria in it, not just magnetotactic bacteria. So how do we find them specifically? And so here we try to get them to go to one place and see if they end up there. And so you take essentially you have a little setup, and I think there will be a picture to show how you would do that setup or how I do it. We basically want to raise our microscope cover slip, have a drop of water on it, and flip it around. So now we have a hanging drop of water. And now we put a magnet next to the microscope, which will tell the magnetotactic bacteria to just swim left and right. And if they swim left and right, eventually they hit the edge of the water drop. And so now with my microscope, I go to the edge of the water drop and I look for some concentration of bacteria. And I can really show their magnetic or magnetotactic by flipping that magnet around on the microscope slide or removing it, and they'll swim away or they'll follow the magnet. So that's how you see them. Uh, it is relatively easy to find them, I think, once you get the hang of it. But I think the tricky thing is that you need that microscope.
Yes.
That's the
Yes.
tricky thing. So it's not yet that easy to do it at home, I think.
And I do worry that the home microscopes, despite a lot of advances, just really aren't good at seeing things at the bacterial level as well
Right.
they might be able to eventually.
Yes, I agree with you.
But certainly I've used your approach and I've loved it.
Yes, yeah, it's easy to find them. I mean, there are other fun tricks we can do, and if there's a link to my website, we have a few videos. Once you have the cultured bacteria, uh, you can do fun things like there you can see that when the cells settle to the bottom of the tube, it's actually a very dark brown black pellet. Because of the magnets that the cells are making, it changes the color of the cell pellet. And then you can put a magnet next to the tube and pull all the cells to them because they're magnetic, right?
Well, this has been really great. And I think that this is something that everyone can really appreciate hearing about. But I'd like to hear more about some of the things that you're doing in your lab if I could.
Sure, of course. Yeah. So, what our interest, my background, as I said, was in cell biology. And so, when I heard organelles, I immediately went to, well, how do they make a compartment? Because it's not an easy thing to do. And if they're going to make a magnet, it's going to require specific types of proteins to concentrate iron, for example. So, how did those proteins know to get to the compartment? And what are those proteins even? So, at the time that I started, we didn't really have an idea as a field about uh, what are all the components needed to make this magnet. The magnet with the compartment that it's in, we call that a magnetosome. So, how do you make a magnetosome? And so a lot of our work over the years has been to set up basically genetic screens to be able to find the components involved in this process. And so a lot of fun. This is the fun type of genetics because you look for cells that are not magnetic anymore. So it's actually kind of really fun to do those screens to find the mutant bacteria. And this is not just me. This is we have a whole very active, vibrant field of scientists studying magnetotactic bacteria. And as a group, we've learned that there's something between 30 or 35 genes that are involved in making the compartment and allowing a magnet to form in that compartment. They don't take the magnets from the outside, right? They bring in sort of single atoms of iron and put them into a compartment and concentrate them and turn them into a crystal. So they're really building a crystal within the cell. You might wonder how a bacterium can build a whole crystal, a solid crystal of an iron oxide inside of the cell. I think I mentioned that they're not bringing the iron oxide from the outside. So how does iron and oxygen come together within the cell? And what we know through chemical experiments that people have done is that the oxygen comes from water. So the oxygen is not from molecular oxygen. The iron comes from outside. It can be in the ferrous form or the ferric form, so iron plus 2 or iron plus 3. And bacteria, all bacteria have many different ways of bringing in iron. And magnetotactic bacteria, depending on the organism, can bring it in somehow. So we think the path might be to go from the single atom of iron not attached to the oxygen, mix it all the way into the cell, and then there are specific proteins on that magnetism membrane that bring it into the magnetism. And if you get enough of it concentrated in there, it passes a critical concentration, just like the sugar in your honey crystallizes. If it's concentrated enough and it starts a nucleus, a little tiny bit of iron oxide that can grow. But this is very simple. What I said is actually way more complicated than that, because magnetite is iron oxide, but the iron is in two different forms in magnetite. It's both iron plus 2 and iron plus 3. And so somewhere, some of the iron either has to get oxidized or some of the iron has to get reduced. And those are things that we don't, as a field, don't really have a good grasp of yet. There are proteins inside of that compartment that potentially have the activity to change the redox state of the magnetic particle, but we don't really know that yet, exactly how they work. And some of the iron might come in through a different way. There's some experiments to suggest that. I would say, if I was going to, it's hard for me to bet which way it is exactly. Is it coming in from the periplasmic space or the cytoplasmic space? It does seem like proteins that are transporters of iron, their job is to move iron across membranes, are involved in this process, specifically within that compartment. Then I think one of the questions you might wonder is, how does it decide to stop? And in fact, how does it decide to be a certain size? Because we did say that these are single stable magnets, but one fascinating thing is that it's dependent on the size of the magnet. So when the magnets are small, less than 30 nanometers, it doesn't have a stable magnetic dipole moment. That thing is just flipping around. So it's useless to the bacteria for orienting in a magnetic field. It's only when it goes past 30 nanometers that that magnetic dipole gets fixed and is not easy to change. And so one answer to the question of how does it know when to stop is that the boundaries of the compartment are as far as it can grow. And so the compartment is about the final size of the magnet. Where it becomes more confusing is when you look at that diversity in the tree of life. Some of them make magnets that are tooth-shaped and elongated, and we don't find compartments that shape really. So those might do it slightly differently. They might start off the process in a compartment and then release the magnet, and somehow proteins shape it as it keeps growing into a tooth shape. And we have a lot of interest in identifying those proteins because I think it can have a lot of potential uses in applications. And so then from the cell biology perspective, we're also interested in how do you make a chain? Because you might think, oh, that's so easy. You have magnets, they are attracted to each other and you make a chain. But in fact, even without making a magnet, they make a chain of the compartments. So it's not magnetic interactions necessarily that make the chain. It's actually proteins that make sort of long ropes or filaments and make sure that the factories to make the magnets are all lined up together so that once you start making the magnets, you make a chain of them. So that's been really interesting too, to figure out how those proteins work. I have a really fascinating picture of what it's like to be inside of a magnetotactic bacterium. Imagine you've crawled in the bacterium and you're standing on the cell membrane looking at the magnetism. This is from essentially collecting many different electron microscopy images of a single magnetotactic bacterium from different orientations and reconstructing it into a three-dimensional image. And when we're looking at it, if you're standing there looking at this sort of bubbles going across, what you see is that the bubbles are coming up from the cell membrane. Because remember, we're sitting on top of the inside of the cell, on top of the cell membrane. And so those blue bubbles are the compartment, the lipid membrane of the magnetism. The red inside is the crystal of magnetite that's being built. And what's really cool is those little rods, yellow rods that are going along that chain. Those are made of a protein called MAMK, and MAMK is a homolog of actin, eukaryotic actin. And MAMK is used to bring those compartments together in a chain so that they're inside of the cell in a chain. They're centered in the cell, so when the cell divides, each daughter gets an equal share of the magnetisms. So, we've gone from essentially, instead of just going to Strawberry Creek in our campus or, any pond or lake, actually have one organism that we can study in the lab as a model, develop tools to find the genes involved and how the magnets are made, and then now study those genes individually. So that's one, I think, continuing aspect of our work. The other one is that when you go to these different places, you notice that there are very different types of magnetotactic bacteria. And they make different shaped magnets, for example. So that's really cool. Some of them make tooth-shaped magnets instead of square-shaped magnets. And how do you control the shape of them? We don't know. And why is it that magnetotactic bacteria are kind of speckled all around the bacterial tree of life? Is it that it's just something that evolved multiple times independently, or do they all share a common way of doing it? And the studies of the model organism, Magnetospirillum, helped us go and look at uncultured magnetotactic bacteria. Again, I mean our field of study. And what people have found is that those genes, some of those genes, a handful of those 35 genes that are involved in Magnetospirillum, are found in all magnetotactic bacteria. So it does seem to be sort of a common mechanism that's elaborated in each different type with other genes added onto it. And there's some very provocative speculation that a lot of this diverse inheritance of magnetisome genes is not through genes jumping around. It's that the last common ancestor of all these bacteria had those genes, and most modern bacteria have lost them. So if we travel back 3 billion years in time, a lot of bacteria would have had some number of magnetisome genes and potentially made magnetisms.
This is fascinating
Right.
because, as I tell students, oxygen's toxic,
Oxygen is
and we
toxic,
don't
right?
we don't think that way because we have, I don't know, 7, 8 different detoxification mechanisms for
Right.
it. But I had not heard about this. This is awesome.
Right. And all magnetotactic bacteria are either extremely microaerobic or anaerobic bacteria, or at least the magnets are made under those conditions. So it also makes sense from the history of it, evolutionarily tracing it, and where they are today. And that's the other fascinating thing, that even before you know my introduction to this topic, you said 1975 is when they were discovered, right? I heard about this in 2000. So there's a lot of work already on this topic. And a lot of that work was to show that, when these bacteria die, they sink to the bottom of the sediment. And over time, all the organic matter is just dissolved and is lost. But this very unique chain of magnets that they've made sticks around. and it can get fossilized. So people search for these magnetofossils, and now we know that there's magnetofossils in even potentially a couple of billion-year-old sediments, if not older, and
Oh, I
possibly
love
even
this.
in for Doc Martians if they're interested in Martian meteorites. There's magnetic particles, and there's speculation that they might be of microbial origin.
Oh, you don't want to get me started on that
Yeah.
subject.
Yes.
But boy, that one, I mean, I'm excited hearing, there's a reason I say hashtag OMG,
Yes. Yeah.
overwhelming microbial greatness, and hashtag WTM, what the microbe?
Right.
Because that's why I love microbiology right there.
Right. Yeah. For me, like I came in so focused on this idea of like an organelle, right? And it's been just an incredible education just from my own, personal introduction to all types of microbes. So, I didn't know anything about microbial diversity in metabolisms. What do people think about the history of life as it evolved on Earth? None of my education taught me any of that, right? And as I've started learning about this topic, and I was fortunate that at the time that I started working on magnetotactic bacteria, most of the people who worked on them were geologists or geobiologists
Right.
fascinated by this, their use as a biomarker. And so I learned a lot about microbes in general being in that context. So I was in Diane Newman's lab as a postdoc. She is the one who introduced me to this topic, and it was every day was learning something new about some interesting microbial metabolism that I didn't know about. And so now I care about the organelles a lot still, but I care about the entire, you know what they could be doing in the environment. For example, there's a lot of speculation that there's so much in some places there's so much magnetotactic bacteria. When they take up iron to make their magnets, that iron is no longer available for other organisms, and iron is an essential element. So there is speculation that they could be taking up potentially up to 50% of the iron that comes into their environment, in the oceans. And what would happen if there were no magnetotactic bacteria around? Would you have an abundance of iron in the ocean, leading to a lot of growth of algae and other things, right? Which could have pretty drastic consequences. So they might be playing a role in the environment that we have not yet really discovered.
Well, that's why my first tattoo, always it was Avite Pawele Domini, all hail the small masters, because there it is.
Yeah.
This is amazing. So as we're starting to close down time-wise, you wanna talk a little bit about your lab group? Because I always like hearing about them.
Yeah, my group, obviously it changes a lot over time. I'm very fortunate to have had really incredible graduate students and postdocs, but also undergraduates at Berkeley, where I think a lot of their uh, unique access to being able to do research and be a part of it. And classes are very big, so you may not get that sort of more intimate, hands-on experience of working with microbes or doing research. So they've been really fundamental in moving the research forward. Uh, I think there will be a couple of pictures up. One of them I have is just to highlight that our field of magnetotactic bacteria is incredibly collaborative and friendly, and incredibly international. In sort of the early days of people becoming interested in this topic, there was a lot of sort of microbial missionary work, of people going around and telling people about it, and there's sort of seeds planted in different parts of the world. And so now we have colleagues in Brazil, in Japan, in China, in France, Germany, and we one of the benefits is that we do a conference every other year on magnetotactic bacteria, hosted by a lab somewhere. And so we as a group end up visiting many different fascinating places. So I think there'll be a picture of my lab when that in 2014 the conference was in Rio in Brazil, and that was really incredible and fascinating. And the people in Brazil, like the researchers there's a lot of exciting things I've worked on, but one of the really cool types of magnetotactic bacteria are multicellular magnetotactic bacteria that are just sort of balls or little football shapes, clumps of cells that don't have a single cell life cycle. They always exist as this aggregate. And every cell in that aggregate has a magnetism chain. So the entire thing swims along a magnetic field,
It's
right? And
just
so they've been
amazing.
studying those incredible, difficult work of enriching them from environments, not being able to culture them, but still learning so much from them. So
Oh,
we get to go and meet people from all over the world and share our fascination. And not only people from different parts of the world, but people with very different interests, ranging from medical imaging all the way to geobiology, come to these conferences and we get to learn from each other.
I guess you could say it attracts the right kind of people.
Yep, it does. Mark, also, I wanted to say I'm very inspired by all the work that you do and how you kind of combine learning with also just art and other things, as we just saw even at the intro. So for a few years now, I've been teaching a freshman seminar class. And this is just because freshmen come in at Berkeley, giant classes. They don't get to really
Hmm.
pursue their interest in science with a small group until much later. So we do different topics, but I thought the best way to do it is hands-on. So for
I
one
agree.
hour a week, we just do every week we meet and we do some kind of microbiology based experiment. Um, because of my interest, microscopy is a big part of it. We started when a grassy columns and we followed it up. But from everything that you always talk about, I thought, we don't want to just make it about facts and science. We also want to make it fun. So they do an art project at the end based on it, something they've learned. And then just is so really just fascinating seeing, um, for example, scraping your teeth to look under the microscope and hearing oohs and ahs around the room. The students are still, they're not jaded by
No.
the realities of life and career and other things. And so they're just seeing the pure joy of learning about microbes has really been exciting. But my inspiration was learning from you to do that.
Well, we should talk. I've got ideas.
Great. Yeah, I would
Um,
love to have more exercises incorporated.
oh, I'll find an excuse to come visit you guys.
Yes.
I'd like to do that.
Yeah, we were going to
Uh,
do that right before the pandemic. We were going to have you
that's what
come and
happened.
right. Yeah.
So as we start to close out, I often ask people what is the one thing that sold them on microbiology, the one observation, the one example, and what is it with you?
Well, I mean, thinking about myself as a microbiologist, the way I think of microbiologists now is that moment where someone said, "Oh, I was talking to Professor Mel Simon and Diane Newman about doing a postdoc in their lab, and Mel Simon was just excited about all kinds of bacteria. He'd started a company called Diversa at the time that were looking at just fascinating microbes that made fascinating products, and he was just listing off different things microbes do, and he said the phrase, "And there are even some bacteria that make organelles." So that, the modern microbiologist that I am came from that. But I thought about your question more. As an undergrad, I was interested in doing research. I knew I wanted to do research. So as a freshman, I thought, how do I find a lab? And this is before websites and having lists of stuff like that. So I had a paper listing of the labs in the biology department at MIT, and I just started calling people. I got to F, and this professor asked me to come in, Maury Fox, who worked on the problem at the time of adaptive mutation in E. coli.
Right.
Would E. coli choose a mutation based on the condition they experience? And that was the question they were working on, which was really fascinating. And so in his lab, I did basically just plate a lot and count. That's it. And then you could learn some really incredible things that way. It was all E. coli. And I never thought of it as microbiology. But I think that's what sold me on eventually microbes being so incredible and powerful to work with. And so I didn't get trained as a microbiologist until I was a postdoc, but I think the
Mhm.
seed was planted when I worked on adaptive mutations in E. coli.
Do you mean the spore was dispersed?
The spore was dispersed. Yes. Yeah.
Well, it has been such a pleasure chatting with you, and I really appreciate your time. You've given me so much to think about, and I'm going to be putting up a flurry of links in the
Thank
show
you.
notes.
Yes.
I may ask your help for some things too, for
Sure.
some of those references as well. But it has been such a pleasure, and I want to wish you and your students and your family the very, very best.
Thank you, Mark. Thank you. It's been great talking to you, and hopefully we can see each other in person at some time in the near future.
That would mean a lot to me.
Yes.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes, with as always awesome links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Dr. Arash Komeili can be found in the Department of Plant and Microbial Sciences at UC Berkeley. Many thanks to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've enjoyed being part of our quality quorum today. See you next week on Matters Microbial.